Good day, everyone. My name is Nur Adlina binti Zainal Abidin, and I am part of the Master in Molecular Medicine ODL program from IMU. Today, I will be presenting of my topic as part of the Molecular Basis of Diseases 1 module, which is targeting the LRRK2 gene as a potential treatment for Parkinson's disease. Firstly, we will start with an introduction to neurodegenerative disorder, which is known as a chronic progressive condition that mainly targets the neuron cells in the nervous system, leaving the neuron cells impaired or damaged. Parkinson's disease, with the short form of PD, is the second most common neurodegenerative disorder among the population worldwide. Mainly, PD can be classified into two categories, which is familial and sporadic based on their origin of cause. Around 10 to 15% of cases of PD are known to be familial as they have a strong family history being associated with the disease, while more than 50% of the cases are sporadic, which arises from genetic alterations that are known to be susceptible to development of PD. Besides the origin of cause, PD can also be categorized into late onset disease, early onset, and juvenile PD, depending on the age of the individual being diagnosed. However, commonly PD is known as a middle-aged or elderly disease and is identified clinically by three motor symptoms, such as resting tremors, rigidity, bradykinesia, and postural instability, in addition to an increasing risk of developing dementia. The presence of Lewy bodies in the brain are the hallmarks of PD, with various cell loss in different regions of the brain. However, there are some other key pathophysiology of PD. PD is characterized by the loss of dopaminergic neurons found in the substantia nigra pars compacta, known as SNPC, which is part of the midbrain, where it is known as the major dopaminergic nucleus of the brain. Studies have shown that many symptoms of PD are known to surface when death of the neurons in the SNPC occurs or becomes impaired. These cells play a role in the production of dopamine, which is a chemical messenger within the brain that helps to transmit signals through the nervous system for regulating movement. The pathological hallmark of PD is the presence of abnormal cytoplasmic deposits within the neuronal cell bodies, which are immunoreactive for the protein alpha-synuclein. These pathological alpha-synuclein protein aggregates are called Lewy bodies and are often accompanied by dystrophic neurites called Lewy neurites. Lewy bodies are often described as a eosinophilic cytoplasmic inclusions that have a fibrillar structure and a dense core, surrounded by a halo as seen in the diagram on the right. The primary structural component of LB is filamentous alpha-synuclein, a protein ubiquitously expressed in the brain. One factor that may be contributing to this phenomenon might be aging, and it has been well established that abnormal protein accumulation can occur as one progresses in age. Mitochondrial dysfunction have also shown to play a role in the pathogenesis of PD, where a vital component of the electron transport chain called mitochondrial complex 1 was discovered to be deficient in individuals with PD. Defects in the mitochondrial complex 1 have shown to be part of driving dopaminergic neurons to cell death because of energy depletion. In addition to that, alpha-synuclein aggregates may also interfere with the mitochondrial function by interacting with mitochondrial membrane, resulting in accumulation of protein aggregates inside the organelle itself, thus disrupting the mitochondrial complex 1 function and leading to an increase in production of reactive oxygen species ROS, which overall may result in oxidative stress. Dopaminergic neurons are known to be particularly vulnerable towards oxidative stress due to the brain consuming high levels of oxygen for the dopamine metabolism, which will generate ROS. ROS, without being neutralized, will bring damage to the cellular structures, which will accelerate neurodegeneration. Neuroinflammation is another process known to contribute to the death of dopaminergic neurons in the brain. The reason because studies have shown that chronic inflammation that is mediated by microglia and astrocytes in the brain plays a significant role in the pathogenesis of PD by contributing to the degeneration of SNPC cells as inflammatory cytokines and chemokines released by these glial cells contribute to neuronal damage, hence causing the disease to accelerate progression in motor system degeneration and impair cognitive function. There are also other pathways that are also known to contribute to the progression of the disease, with several studies suggesting the abnormal protein clearance in the protein misfolded clearance pathway, which are the autophagy lysosomal pathway, ubiquitin proteasome system, as well as the cellular energy metabolism. Autophagy is a cellular process that degrades the recycles damaged organelles and proteins. However, mutations in genes like LRRK2 and GBA disrupt this pathway, leading to an accumulation of toxic materials. The dysregulation of autophagy then results in neuronal damage in PD, and this may prompt neuroinflammation inside the brain to occur as well. UPS, on the other hand, is responsible for degrading misfolded and damaged proteins. Dysfunction in the system is partly due to mutation in genes like PARK2, leading to the accumulation of toxic proteins as well, including alpha-synuclein. An impaired protein clearance system can further well contribute to the formation of Lewy bodies and neuronal death, hence accelerating the progression of PD. While disruption in the cellular energy metabolism, on the other hand, is usually pointed to the interference occurring in the cell's powerhouse organelle, namely the mitochondria, which often in PD individuals arises due to mutations in genes like PINK1 and PARK2, leading to impaired energy metabolism and increase in oxidative stress. In addition to that, neuron cells with their high energy demands are particularly susceptible to disruptions in mitochondrial function and is highly affected. According to the Genome-Wide Association studies, here are a few genetic alterations known to be susceptible to the development of PD. SNCA, which is alpha-synuclein, are known to integrate presynaptic signaling and membrane trafficking. However, defects in this gene leads to misfolded aggregate proteins being produced. Leucine-rich repeat kinase LRRK2, on the other hand, is known to be involved in multiple cellular processes such as vesicular trafficking, autophagy, and cytoskeletal dynamics. Alterations in this protein is known to be contributing to the progression of PD. Part 2 plays a critical role in ubiquitination, the process whereby molecules are conveniently labeled by ubiquitin and directed towards degradation in proteasomes or lysosomes. Defects in this gene results in faulty UPS system pathway, thus contributing to the development of PD. Part 7 is a gene that encodes the DJ1 protein, which helps in protecting neuron cells against oxidative stress and cell death. The PINK1 gene encodes the protein named P10-induced putative kinase 1, which is located in the mitochondria and functions to protect the mitochondria from malfunctioning, especially during periods of cellular stress when during an unusual high energy demands. The GBA gene, on the other hand, makes the enzyme called beta-glucocerebrosidase. This enzyme is active in the lysosome structure and helps in degrading misfolded proteins. Defects in this gene will hence interfere with the autophagosome lysosomal pathway. However, in this presentation, we will be focusing on the LRRK2 gene, which is known to be one of the most susceptible genes in the development of PD, next in line to alpha-synuclein SNCA gene. Here are some of the literature reviews that highlighted the roles of LRRK2 in the development and progression of PD, and why targeting this particular gene has potentially high therapeutic value in the treatment of Parkinson's disease. Leucine-rich repeat kinase 2, or known as LRRK2, is a multi-domain protein kinase encoded by the Part 8 gene located within chromosome 12. It is part of the ROCO protein family due to the presence of RAS of complex ROC in the protein domain, which constitutes to the catalytic core of the protein. The kinase protein bears a kinase, GTPase, and scaffolding domain is highlighted on the right diagram. In addition to that, LRRK2 may also act as a scaffolding protein due to the presence of leucine-rich repeats in the N-terminal and the WD40 repeats at the C-terminal, which are vital for protein-protein interaction with the kinase protein. It was discovered that initially, the LRRK2 protein exists as a monomer being stabilized by the protein 14333 in the cytosol. When activated by RAS substrates and recruited, the protein kinase will be localized into the cell membrane to the hydrolysis of GTP, resulting in the dimerization of the protein. Activation of the protein will then recruit other downstream signaling proteins, and this protein is known to be highly associated with the MAPK signaling pathway. Why target the LRRK2 gene? This is because it is one of the most common genetic alterations in familial and sporadic PD. It has also been discovered to be expressed in multiple cells such as the brain, the kidney, the heart, and the lung. They are also highly expressed in immune cells such as the microglia, which are important immune cells found in the brain. LRRK2 are also involved in multiple cellular processes such as vesicular trafficking in the Golgi and ER, autophagy, and cytoskeletal dynamics. Common alterations found in the LRRK2 genes such as G2019S and R1441C are known to be result in the hyperactivation of the kinase activity, which may lead to cellular dysfunction and neuronal cell death. The autophagy lysosomal pathway can be categorized into the macroautophagy, mitophagy, and chaperone-mediated autophagy. In macroautophagy, mutants LRRK2 have shown evidence of decreasing the levels of LC3 and LAMP1, which leads to the repression of fusion between autophagosome and lysosomes, resulting in large stranded dysfunctional autophagic vacuoles occurring in various regions of the cell. LRRK2 are also known to be highly associated with chaperone-mediated autophagy. In the CMA process, chaperones proteins such as heat shock 70 are required to recognize the target substrate, and this chaperone binds to the lysosome protein LAMP2A to internalize substrates which are then degraded by a lysosomal protease. CMA is largely responsible for alpha-synuclein clearance since alpha-synuclein clearance contains a recognition motif and can be recognized by the chaperone. However, it was reported that in mutants with LRRK2 G2019S, it acts on the LAMP2A and interferes with the CMA process, thus decreasing lysosomal degradation of the protein, resulting in precipitation and accumulation of alpha-synuclein in neuronal cells. Furthermore, it was reported that the mutant LRRK2 which carry alterations such as G2019S and R1441C in particular are susceptible to autophagosome lysosomal pathway and have shown to decrease autophagic flux in cell lines such as microglia and astrocytes. Conceivably, impaired lysosomal function will result in deficient protein clearance and could fit the model of Lewy body pathology where there is an accumulation of aggregation of alpha-synuclein proteins. Neuroinflammation, on the other hand, is the process that requires immune cells in the brain to release inflammatory cytokines, which results in the recruitment of peripheral immune cells. Scientists have discovered that inflammation induced by liposaccharides LPS will lead to an increased expression of LRRK2 in microglial cells, which will then prompt activation of signaling pathways such as nuclear factor kappa beta, resulting in an increased secretion of pro-inflammatory cytokines. The release of these pro-inflammatory molecules from activated microglia into the brain will enhance the oxidative stress of the SNPC, which will then result in the degeneration of dopaminergic neurons, as a lot of studies reported that chronic inflammation can contribute to the generation of dopaminergic neurons and the progression of PD. Evidence have also shown that mutant LRRK2 with mutations R1441G may also force microglia towards a pro-inflammatory state, which in turn promotes neuroinflammation and resulting in neurodegeneration through an increased expression of pro-inflammatory cytokines. Studies have also indicated that mutants LRRK2 such as G2019S that results to an overphosphorylation of downstream MAPK signaling pathway may also lead to activation of neural death signaling pathway through the JNK CGN signaling pathway, which further highlights that LRRK2 may be involved in the innate immune system of the cells as well. Now, it is most important to emphasize that existing PD treatments only elevate symptoms and do not delay or halt progression of the disease, and current therapeutic approaches for PD aim to restore motor function by re-establishing dopamine signaling. Hence, scientists turn to LRRK2 as a potential therapy for the progression of PD. Currently, there are two therapies undergoing clinical trial that are targeting the LRRK2 gene. The first is a small kinase inhibitor DNL151, which action of mechanism are ATP competitive. It is consumed orally and can be delivered through the blood-brain barrier. In the first clinical trial, the recruited participants are all in early stages of PD with some kind of LRRK2 alterations. In December 2017, phase 1 of the clinical trial showed tolerable toxicity profile and safe to be consumed, and have also showed evidence of slowing down the progression of PD. These results then forged way for the second phase of the clinical trial, which began in May of 2022. Another therapy strategy for targeting LRRK2 is the antisense RNA molecule BIIB094, which is an antisense oligonucleotide where it binds to the mRNA for LRRK2 via base pair complementary and mediates its degradation, thus lowering the expression of the LRRK2 in the cell. Phase 1 of the clinical trial started in August of 2019 using the intra-athecal injection directly into the cerebral spinal fluid. This method has also been used to treat Huntington disease and have showed decrease in mutant genetic expression and correspondingly improved the clinical symptoms of individuals suffering from Huntington disease. And hopefully this will be the case as well for patients that are suffering from PD. Some challenges that have often arisen in targeting LRRK2 as a treatment for PD is that further studies of LRRK2 is required in order to identify novel molecular targets that target LRRK2 dependent in the neurodegeneration of PD. The reason for this is because of the heterogeneity shown by the LRRK2 gene, as different mutations shown in the LRRK2 will have different distinct cellular downstream effects. In addition to that, the drug delivery method for small kinase inhibitors targeting LRRK2 is crucial, as most of the compound inhibitors that are known to be effective against the gene in PD have shown to be unable to penetrate the blood-brain barrier. Besides that, most of the therapy targeting LRRK2 have shown similar side effects of enhancing respiratory problems, as LRRK2 protein are also being expressed in the lungs. As how this came to be, it still remains unclear, however. There is also a need for new non-invasive biomarkers for accurate detection of LRRK2 activity and for monitoring purposes in the progression of PD from the early stages. Currently, there are two types of biomarkers which are used for serving such purposes, which is BMP, a phospholipid that are considered specific to lysosomes, and phosphorylation of RAP10 and LRRK2 phosphorylation on the serine 935 LRR domain site in the peripheral blood mononuclear cells and whole blood. Currently, small molecular kinase inhibitors targeting LRRK2 is showing a promising future as new therapeutic strategy for individuals with PD. However, there are also other wide range of therapeutic strategies aiming to treat both LRRK2 and PD by reducing the function of this enigmatic protein, such as LRRK2 dimers, which are believed to have greater kinase activity than LRRK2 in their monomer state, and disruption of protein-protein interaction that require for pathogenic effects of LRRK2. In conclusion, therefore, with so many routes to achieve LRRK2 inhibition being taken, the prospects for developing new PD medication based around targeting LRRK2 are looking promising. Where lead molecules and different strategies fall by the wayside, there are still plenty of alternatives to take over. However, scientists are also on a mission to have a much better complete understanding of the LRRK2 and its mechanism of action for further identification of novel targets in treating PD and better future in improvement of medical treatments for patients. Here are some of the references used in this presentation. That's all from me. Thank you and goodbye, everyone.